Chapter 1, Principles of Genetics and Overview. Contents. Genetics has a rich and interesting history. So, genetics is a study of genes. Inheritance is how traits or characteristics are passed from generation to generation. Chromosomes are made of genes, which are made of DNA, and genetic material are found inside of the nucleus. Gregor Mendel is considered the father of genetics. So, history of genetics, 1600 to 1850, there is Genomics. So, William Harvey proposed the theory of epigenesis. So, structures such as body organs, they are not initially present in the early embryo, they are formed later. Schleiden and Schwann proposed cell theory, 1830. All organisms are composed by basic structures called cells. So, William Harvey, he is the world's greatest creation scientist, one of the world's greatest. First fully described the circulatory system with mouth, vein, and the heart. He is the personal physician to King James I, and he is also the pioneer of modern embryology. So, according to the people on the era, believe in pre-formation. In pre-formation, it is believed that in the sperm there will be a small mini human called homunculus that will grow into a regular sized human, meaning that the ovum would just be a receptacle and doesn't have any value other than to receive that sperm. So Harvey is opposed to the pre-formationist. The Origin of Species, 1859, it is published by Darwin, and according to the Origin of Species, it is something that proposed descent with modification. So, descent with modification means we have ancestors bringing parents, parents bringing sons, and then sons bringing granddaughters. Each one of these succession will have a little bit of modification happening in each generation, and then from these ancestors, new species will arise. Next will be natural selection. It is the mechanism of evolutionary change and also the theory of evolution. So around the same time, Alfred Russel Wallace also proposed this theory of evolution. So Gregor Mendel, he is an Austrian monk who experimented with pea plants and used pea plants because they are available, reproduce quickly, and have differences in traits. So he understood that there's something that the pea plants carry from parents to the offspring and to a generation. He called it factor. So Mendel, in mid-1800s, he performed countless experiments, around 1,000 something experiments, to show that there is something that is carried from the parent to offspring. So he was the first to apply the experimental approach to question inheritance. For 7 years, he bred pea plants and recorded the inheritance in the offspring. So he came up with the particulate hypothesis of inheritance, in that parents pass on the offspring separate and distinct factors from themselves into the offspring. So we call it genes now, and they are responsible for the inherited traits. Okay, so rationale of inheritance. 1866, Mendel publishes his findings, and he worked with peas and used quantitative methods to promote ideas. Traits are passed from generation to generation and from parents to offspring. So he is fortunate that he chose the garden pea, because the garden pea has many varieties, and you can be able to control how you breed them. You can just cut off their anther, then you can use a brush to transfer a pollen to the pollen of another flower. And the traits of pea will be distinct and clearly contrasting. In terms of particular hypothesis, Mendel crossed true breeding plants which has two distinct and contrasting traits, for example, purple or white flowers. So to prevent self-fertilization, he cut off the immature stamens. Stamens are the structure that produce pollen. And he dusted the carpel, which is the egg producing structure similar to ovum, from another plant, which is opposite of the plant that he wants to study. So the couple matured into pods containing seeds, which we call peas. Once planted, the seeds grow into offspring plant. So in the pea generation of true breeding parents, you have purple flowers and white flowers. And when they are crossed, they only turn out with purple flowers, meaning that the offspring are all purple. But in the after generation, only 3 of them are purple, but 1 is white. So in a cross called the monohybrid cross, Mendel cross-fertilized true breeding plants that were different in just one character, which is mono, in this case flower color. So he allowed the F1 or hybrid generation to self-fertilize. You have P generation, parental. F1 generation is filial one, or the first generation, and then all of these first generations will breed with each other. Then you have the F2 generation, which is second filial generation. From here, you can see that there is a ratio of 3 to 1. Mendel studied 7 characteristics in the garden pea. Flower color, purple or white. Flower position, axial terminal. Seed color, yellow or green. Seed shape, brown or wrinkled. Pod shape, very opposite. Pod color, green or yellow. And stem length, tall or dwarf. So, by breeding these opposite or contrasting traits together, we found out that the ratio would always be near to 3 to 1. One trait will be 3 times as much as the other. So in this case, flower color, purple has 3 times of the offspring compared to white. Axial is the more than terminal. Yellow is 3 times more than green. Brown is 3 times more than wrinkled. And then same will to inflated, green and tall. So next will be mitosis and meiosis. Mitosis will be the division of cells without reducing number. So to create a daughter cell, which is 2 diploid cells. Meiosis will be where you have a cell divided into 4. In this division to 4, they actually will lose half of their chromosomes, meaning that they only have half chromosomes compared to the original. So we call it haploid. So most of eukaryotes have a characteristic number of chromosomes called the diploid number. So this is a chromosome of a human male, 46XY. So each one of the chromosomes would have its pair 1 until pair 2. But for the last pair of chromosomes, you have X and Y. Chromosomal theory of inheritance. Inherited traits are controlled by genes residing in chromosomes, and genes are transmitted to gametes. So it means that there will be a genetic continuity from generation to generation. Alleles. Mutation produces alleles of a gene, which is a source of genetic variation. Genotype is the alleles for a given trait. Phenotype will be the expression of genotype, which is the observable trait for the genotype itself. DNA is the carrier of genetic information of protein. The research of Avery, MacLeod and McCarthy in 1944 published experiments showing DNA was the carrier of genetic information in material. The DNA is antiparallel, double-stranded helix and made out of nucleotides. So DNA is a polymer. It has a chain of many smaller units called a monomer. So each monomer has a nucleotide consisting of a sugar, bonded to phosphate, bonded to bases A, T, C, G, adenine, cytosine, thymine and guanine. So nucleotides form A, T and GC, in that A, adenine can only pair with thymine, guanine can only be pairing with cytosine. So we call it a complementary base pairing from the helix. Okay. So RNA is similar to DNA except it is single-stranded. It has uracil instead of thymine, and sugar RNA is ribose instead of deoxyribose. So this is the central dogma of genetics. DNA to RNA to protein. DNA has to be transcribed to RNA, and then RNA needs to be translated into protein. Codons. Codons are triplet nucleotides present in mRNA. It is the genetic code. Each triplet encodes for insertion of a specific amino acid into a growing protein chain. Proteins, they are the end product of gene expression. Protein action or location in a cell produces phenotypes. So we have 20%. So we have 20 different amino acids, but the number of combinations of these 20 would allow a lot of diversity. So we have first letter UCAG, second letter also UCAG. Let's say that our protein has a codon of UUC. So if UUC, our amino acid will be phenylalanine. What if our codon reads AUA? Okay, so if it is AUA, then it is methionine. Resistant enzymes. In 1970s, researchers discovered that resistant enzymes in bacteria cut viral DNA at specific sites. Resistant enzymes allow the advance of recombinant DNA and cloning. So this is the first clone that we humans have made. Dolly, which is a female domestic sheep, cloned from another adult somatic cell using a process of nuclear transfer. Okay, so you have a donor egg, and then you remove its nucleus. So you have a nucleus without an egg. So this egg can accept a nucleus from another organism or another cell. So you put an egg's nucleus and fuse it with electricity. So now you have egg which is fused with the cell. So now if you allow this cell to develop into an embryo, it will be just like the actual original cell, where you will be just like the original cell where the donator has been donated. So what will it look like? As you can see here, the black-faced sheep only gives the egg but no nucleus. The nucleus comes from the white-faced sheep, meaning that this white-faced sheep will actually have a clone itself being born. The pair of biotechnology is continually expanding. Biotech is now used in healthcare, supermarket products, agriculture, and also the cont system. In agriculture, we use it to modify genetically on plants, on increase herbicide, insect and viral resistance, nutritional enhancement, water use reduction. So you can see here, these are all the countries that create biotech crops, meaning that they are crops which genes have been tampered with. Section 1.5, we have biotech in genetics and medicine. So in genetic medicine, we actually look at somebody's genetic to forecast what kind of disease that baby will be born with, and we can also forecast the disorder heritability. How well can it translate from one generation to another generation? So these are all of the DNA tests currently available for young couples. Genomics, proteomics, and bioinformatics are new and expanding fields. Genomics, we study the function, structure, and evolution of genes and genomes. Proteomics, it is like genomics but about protein. Bioinformatics, if you look at the name informatics, you are using IT. Bioinformatics means that you are using software and hardware for processing data of nucleotide and protein, which is the bio part, which is the life part. So life has common origin. Genes with similar function in different organisms are similar in structure in DNA sequence. So modern approaches to understanding gene function is by either liver genetics or gene knockout. We said that we have a gene that we don't know. Right? So we try to use a DNA sequence called a gene knockout. We shut off a gene one by one to see what it does. So it's just like you have a lot of switches in your house. You don't know which switch causes which switch is for which light or which fan. So turn it on one by one. Whenever you try that, and then suddenly a lamp turns off, then you know that, oh, this switch is for this lamp, or this switch is for that fan. So model organisms, they are organisms that we use so that they can be tested, and then we can extrapolate the test results to the human being. So model organisms must be easy to grow, short life cycle, produce many offspring, and also genetic analysis will be straightforward, meaning that they are not complicated. Okay, so to study some human diseases, we actually use a lot of organisms, so they will be afflicted with diseases for our benefit. We have E. coli for colon cancer, Staphylococcus cerevisiae for cancer, and Werner syndrome. Drosophila melanogaster, or the fruit fly, is for disorder of nervous system and cancer. C. elegans, Chromosoma elegans, it is a fly, it is for diabetes. E. radio is for cardiovascular disease, and M. musculus is what's called the white mouse. Lunch, Nyhan disease, cirrhosis, cardiac syndrome, and many other diseases. So they work as lab rats. They are actually literally rats that work in lab, which we as humans test on. What of human disease? We use recombinant DNA technology. So recombinant DNA technology is able to transfer DNA across species. So this is the first generation of model organisms. You have the rats, musculus, and then you have the Drosophila melanogaster, which is fruit fly. So model organisms, now we have viruses, including lambda-phages, bacteria, Escherichia coli, and these Saccharomyces cerevisiae. So as you can see in this timeline, genetics is becoming better and better as time passes, and the application will be much more bigger and bigger as time passes. So 1865, Mendel is at the stage to study of genetics. Genetics rapidly developed from Mendel's piece to Human Genome Project. 1962, we have Nobel Prize awarded to Watson and Wilkins, and from them, there's actually many Nobel Prizes given to geneticists. What about the future of genetics? Society is faced with a host of sensitive genetic issues, prenatal testing, ownership of genes, access to and safety of gene therapy.